Good morning, everyone. Welcome to this new Next Friday talk. Today, the speaker is Professor Eleonora Maggioni, a professor at Politecnico, and she will host a talk about multimodal data integration for affective disorder, if I'm not wrong. So, please, I pass you the speech. Thank you.
Thank you so much, Leonardo. Thank you to everyone for being here. So today I will introduce our research activity that is conducted in collaboration between Politecnico and Politecnico of Milan, and specifically the psychiatry unit, where we try to integrate multiple neuroimaging and electrophysiological modalities and data across multiple sites and research networks that involve worldwide centers in the study of affective disorders. Sorry, there is a typo already in the title. So we will start by showing that the potential of different brain imaging and signaling techniques. You will know that there are multiple techniques that are available, and depending on the specific application, we can focus either on one or multiple of them. For instance, very easy to use is the electroencephalography, or it's very, very common magnetic resonance imaging when we need to collect data from both the brain or body structure and function. Or there are other techniques like near-infrared spectroscopy, but each of them has a specific sensitivity and specific spatial or temporal resolutions. And this is why when we try to integrate them, we really have a more comprehensive framework and picture of what we are interested in. And specifically in the study of the brain, what is very powerful is the integration of multimodal magnetic resonance imaging with also EEG that could be performed within the scanner. If you have any questions also during the talk, please feel free to ask. And here is just an overview of the different sensitivities in terms of spatial and temporal resolution of techniques that try to probe brain functional processes. And we usually, of course, act in this area because these are invasive techniques that can go to the level of multiple neurons, like the local field potentials, or the patch clamp, that is something that cannot be performed in humans unless with very invasive strategies. And so we will focus more on this area, that is the one of non-invasive techniques with EEG, functional near-infrared spectroscopy, MRI, multimodal MRI, including specifically functional MRI, but also, for instance, positron emission tomography that gives information on the metabolism. So there are multiple resolutions and multiple information that can be retrieved by these techniques. And in recent years, there has been this diffusion of initiatives and a digitalization of health that is providing new and unprecedented sources of information worldwide, from electronic clinical records, wearables, and different sources. Or we also have many researches where we are having a longitudinal follow-up of the same patients. And so there are multiple initiatives like the randomized controlled trials, clinical trials, or mega biobanks, or initiatives that are collecting genetic data across large populations. And all of them are providing unique sources of data that can be used for research and that are increasingly available also as an open source datasets. And these are some examples of biobanks worldwide. We will focus a bit more on the UK Biobank, where we have, since we have worked on data from this biobank that is centered in the UK, and that is of around 500,000 participants who gave their data longitudinal and for research. And the main, let's say, compromise that has to be found is that usually the number of subjects goes at it's inversely proportional to the phenotypic detail. So many times we have a few subjects that are very well phenotyped, meaning that we have a lot of information from the same subjects, and as soon when we move to greater numerosities, we lose like the detail many times. So we have maybe less information from bigger samples. So we will focus a bit on these two dimensions, as in my research and the research of our group, we have focused a lot on the phenotypic detail by the multimodal integration of the small cohorts by taking as much as information as we could. And then we will show some examples of the multi-site and big network data analysis that are more limited in terms of modalities available, but that are with quicker samples. Okay, starting from my let's say background expertise, we will focus a bit on multimodal data integration. So as I was saying, the magnetic resonance imaging is by far the most powerful non-invasive technique that can probe both, give a multi-perspective view of the human brain, starting from the brain structure using the imaging. And then there is the diffusion-weighted imaging that traces the white matter pathways across the brain. And after a lot of processing, then we have the estimation of the white matter threats. And this can be used to probe and to estimate the structural connectivity within the brain. Whereas the functional MRI technique gives information on the brain oxygenation state through the bold contrast that we will see, and can be used to extract brain activations or functional connectivity. We will focus a bit more on the functional techniques, so starting from fMRI. Usually it is based on the blood oxygenation level dependent contrast that is called BOLD. That is based on the neurovascular coupling hypothesis. So we know that somehow the oxygenation changes that are probed by the BOLD effect are somehow related to the underlying neuronal activity. And how this is possible, because we know that when there is an increase on the neuronal activity within a specific brain region, we have an increase of the metabolism within that region and of the local oxygen consumption due to the metabolism. But there is an increased blood flow that comes to the region that is activated, bringing new oxygen that is in excess compared to the one that is really needed. And this is why the net effect with some delay, that is the hemodynamic delay, is that we have an increase in the oxygenated hemoglobin and a decrease in the deoxygenated hemoglobin around 5-6 seconds after the neuronal activity. This changes the magnetic susceptibility because the BOLD effect is sensitive to the balance between oxygenated and deoxygenated hemoglobin, and we have an increase in the BOLD signal. So we have many fMRI volumes, one volume of the brain of the BOLD contrast every 1 or 2 seconds. And every 2 seconds, we can track the change of the oxygenation change of this BOLD state, this BOLD signal in all the points of the brain. So using fMRI, we can go in the deep cortical and subcortical regions and try to estimate what is happening in terms of neuronal activation, because we have the oxygenation change that is related to the underlying neuronal activation. And this is just an example. We did this work in collaboration with Professor Brambilla from the University of Milan and the Polyclinic of Milan. He is a psychiatrist with whom we are working, and we have the research group that is joint. And we have studied in this case, for instance, the neural correlates of emotional imagery. So we asked the subjects to imagine some emotional or motor verbs, and we tried to extract the brain activations related to this activity. And this has been performed in collaboration with Barbara Tomasino from Udine, so the Friuli Venezia Giulia. And we found that, for instance, emotional imagination was activated the left putamen, insula, mid-cingulate cortex, and right putamen. And we also found that females were different than males in terms of the regional activations, and specifically females had greater activation in the right putamen, for instance. So we try to understand the impact of sex and also age-related changes in these patterns. Just if you are interested, we can go in the detail. So as we were saying, speaking of the fMRI, it's nice to have an integration between fMRI and complementary techniques. As we saw, the bold signal is sensitive to the oxygenation changes within the brain. But these oxygenation changes are only indirectly related to the neuronal activity. And this is why then we also have a very nice spatial resolution. We have an whole brain information, so also subcortical, but we have a temporal resolution that is of around 1 or 2 seconds, if we are lucky 1, but many times 2 or 3 seconds. And we have an indirect link to neuronal activity. And what is the technique that gives us information on neuronal activity that is direct and that can be also integrated with fMRI is the EEG. The EEG records the synchronized electrical potentials merely from large pyramidal neurons with an excellent temporal resolution. So it's like if we track in the real time the neuronal activity continuously with up to 5,000 Hertz of temporal resolution. But of course, as you can see, we cannot, if we are in non-invasive applications, we need to just have information from the scalp, and we have a superficial measure and with a very low spatial resolution. So their advantages and disadvantages are exactly complementary. And for instance, in the EEG, we have the spatial inverse problem for which we cannot localize brain sources with like certain solution from the scalp potentials, whereas in the fMRI, we cannot understand what are the exact timing of the neuronal events that are generated, like the underlying oxygenation changes in the entire brain. So this is why, by combining them simultaneously, we can have a simultaneous neuronal and hemodynamic information with a very better spatial and temporal resolution. So this is the dream, let's say. But when we need to have simultaneous EEG fMRI, we have many problems, like technical problems that we are many times we have fun in trying to, let's say, solve these challenges. First, we need, but this is already implemented. We have MR-compatible EEG sensors and cables that are needed. Then we need also an EEG amplifier that is within the MR scanner room. We also need to fit the EEG cap within the magnetic resonance bore and within the coil that surrounds the subject during the MRI. And then, after all the equipment is fine, we need to guarantee the synchrony between EEG and fMRI, and also remove the artifacts that affect the EEG signal that are very bad, but it's also very, let's say, nice to perform this noise reduction. So this is the standard equipment that we have in polyclinical. So we have the subject with the EEG channels lying in the scanner. Then we have, we also have the stimuli that are presented to the subject to have the task within the scanner while the participant is wearing the EEG cap. Then we have also the EKG, the heart rate that is captured within the scanner through an EKG. Okay, then we have all the synchronization that is needed. So we took years to try to understand how to use these systems, but now we know how to do that. Then there are many artifacts within the EEG. First, there are delta of potential on EEG leads due to the fMRI volumes that are recorded, to the radio frequency pulses. And then we also have a more subtler artifact that is the heart rate related artifact called cardiac pulse artifact. And this pulse artifact is more difficult to be removed. So I spent some months trying to extract a pipeline for the identification and removal of the cardiac pulse artifact that are very difficult to be removed. And at the end, through independent component analysis, we created a sort of semi-automatic pipeline.
I was wondering, for the ECG artifact removal, you were able to do this removal because you already had like a synchronized.
Yes.
Okay, so
exactly.
in the case you don't have the synchronized EEG, you have to deal with the ECG artifact.
Let's say that if you do like the independent component analysis, sometimes in the EEG, even outside the MR environment, inside it's terrible because the pulse artifact is even proportional to the magnetic field. So like a two-track field, you could not even see the signal. But in normal EEG applications outside, when you perform independent component analysis, many times you have like a sort of component that is extracted that is related to the heart rate, and you can find it because it depends a lot on the vessels that are near the channels, and sometimes it can also be that some channels seem an EKG channel even if they are EEG, depending on the position of the vessels. So I would suggest you to perform an ICA if you have enough channels, and then you could find a typical pattern that is the heart rate and mark it. And this is what we did, because even after averaging artifact at the channel level, at the end it was still present, this terrible artifact, so at the end we relied on ICA. But we can interact on that. And then we published finally, like a lot of years ago now, this manuscript that is being used as a reference for the EEG fMRI processing in our group. And finally, we have an EEG that is within the MR and that is also clean. That is not so. Sorry that I moved the slide, but probably
Stop the shadings.
My connection probably. Okay, with Elena Bondi, who did her PhD here in Politecnico in collaboration with the University of Milan, we then worked finally on the different potential perspectives of integration of EEG and fMRI information. So briefly, we can use EEG to drive the fMRI analysis, having the neuronal information, or use them in a symmetric space, using joint independent component analysis, for instance, or use the fMRI to drive the EEG source reconstruction. So there are multiple applications. One of the most useful ones in the clinic is the usage of EEG fMRI for epileptic source identification, because on the EEG we can immediately track the epileptic abnormalities because we have the timing of these events that we recognize on the EEG, but we don't know the underlying sources. So we mark, for instance, in this case, these events, and this work has been performed in the Irs Medea Opposite Parine during my PhD. We use them in the analysis of the fMRI activation. This is the neuronal events. We convolve them for the hemodynamic response, and at the end, in a model-driven way, we extract the also subcortical, cortical, but also deep sources that have a bold signal that is related to the neuronal epileptic events. And in this way, it's a sort of identification without having the deep cortical electrodes that are invasive, of identifying the epileptic areas through EEG fMRI. And we are also using EEG fMRI to study the major depressive disorder in polyclinical. And for instance, in this case, we used event-related potentials that were recorded during the fMRI session to an inhibitory task, go-no-go task. We extracted the event-related potentials, and we did this. We used them within the fMRI activation analysis using a general linear model. And at the end, we extracted the fMRI responses to the latency and amplitude of each ERP peak. So we extracted, for instance, the N200 peak, the P300, and we could extract the brain responses to each of these peaks to try to understand better, like also the flow of information for the stimulus processing. And then we also performed the comparison between these brain regional activations between patients and controls. And in this case, just to be very, say, fast, we found that major depressive disorder individuals had altered brain responses to specific neuronal processes during the inhibitory control task. And this was published recently by Elena on neuroimaging. Then, since we have EKG within the fMRI scanner, the EKG gives us information on the heart rate. And you know that we can also study the heart rate variability and the autonomic nervous system. And having heart rate variability with fMRI in the same time, we can study somehow like how the mental processes and brain processes are related to heart rate variability processes. And we could also study cardiovascular, let's say, comorbidities of mental disorders. This is because depression is many times associated with greater cardiovascular risk. And there are multiple factors that could be, let's say, mediate this interaction. They could be due to therapies. For instance, if they take some therapies, they could have greater cardiovascular risk. But there could be biological factors like autonomic dysregulation that could be somehow the bridge between cardiovascular risk and depressive risk. And this is why with EKG and fMRI, we want to study a bit more this, let's say, this interaction between these two diseases. And this is the subject of the Federica Goffi's PhD, who is the student here in Polytechnical, always with Polyclinical. We study the depressive disorder versus healthy controls in terms of heart rate variability and brain responses. Here we have the fMRI analysis, so we have the activity of the brain. Through the EKG, we extract the heart rate variability changes over time by extracting the sympathetic and parasympathetic system dynamics over time. And we try to study what are the brain responses to the sympathetic and parasympathetic dynamics. And we compare them between depressed individuals and controls. And what we found is that depressed individuals had altered brain responses to vagal and sympathetic activity in regions that are merely located in the hippocampus and the precuneus. And these regions are in the literature known as the regions of the central autonomic network. That is the brain network that controls the autonomic regulation, like the heart rate. So our hypothesis is that since these regions are both involved in depressive symptoms and in autonomic regulation, we think that this could be somehow, let's say, these two networks could be interacting And this could be like a biological reason for the increased cardiovascular problems in depressed individuals and vice versa. And this has just been accepted, this work. Then, since we love to, let's say, have different types of sorry, okay, this so you can see the faces. Okay, with Federica and with Elena Scalbi, who is another during our Master of Science thesis, we integrated heart rate variability with fMRI, as we just see, and also, so we extracted the brain regions involved in heart rate variability, and we used the diffusion tensor imaging to trace the white matter pathways that somehow connect these regions. And this work has enabled us, let's say, to, since we were at ultra-high field, we extracted these fMRI regions of response to vagal and sympathetic systems, and then we tried, let's see if we traced the white matter tracts between the regions of interest with very high resolution. This work was presented at the Metrox Training Conference of the IEEE and won the Best Conference Award, paper award. So I just close this multimodal chapter, let's say, by saying that we have also neural simulation technologies. I don't know if you know these techniques. They are becoming increasingly used also not just in the clinics, but also for cognitive enhancement, for instance. We have multiple non-invasive, but invasive, but also non-invasive techniques like the transcranial direct current stimulation or the TMS, the transcranial magnetic stimulation, that are used to stimulate non-invasively some brain and neuronal activity. And these can be used to somehow drive the analysis of the functional brain processes. For instance, if we stimulate this motor cortex with the TMS, we can have that we move the hand, because we have the stimulation of the motor region, and so we have the movement of the hand that is not controlled by us, controlled by the stimulation. So we use this type of technique while having the EEG recordings to try to understand the propagation of the neuronal stimulation in the brain. So this can be used for investigation purposes, but also for therapeutic purposes, like deep brain stimulation for the Parkinson, and so on. And many times, the ideal framework is to have an MRI of the subject to drive the stimulation of the specific gyri that are of the subject through a neuronavigator system that has the brain of the subject, so we know where to focus with the stimulation. And during this stimulation, we can record the EEG. By doing this, we have the so-called TMS evoked potential. So we try to extract the potential that is evoked by the neural modulation. And we study this cortical excitability mainly. And we use this within the Subbrain project that was in collaboration with the Catholic University and with the Polyclinic of Milan. And in this project, we integrated virtual reality that was validated to induce a specific emotion that is called the sublime. Then we will go into detail if you are interested. And during this VR navigation, we had the EEG recordings. And immediately after, we stimulated the dorsolateral prefrontal cortex because we wanted to know whether these scenarios could be therapeutical for depression. So we stimulated the region that we know is involved in depression to see whether it was modulated by the emotional experience. Okay, this is the sublime introduction that simply this type of emotion is called awe, and is like the combination of two opposite polarities like terror and amazement. And this emotion is considered as potentially therapeutic for depression. And we have tried to recreate it through multicentre experiences and specifically the VR. And this is the experimental protocol, where we have that during the VR navigation, we have 4 virtual reality scenarios. 3 of them are validated to induce O, whereas 1 is neutral of reference. During the navigation, we have the EEG recordings. Immediately after, we have the TMS, that is the transcranial magnetic stimulation during the EEG. And we have emotional questionnaires to try to understand how was the intensity of the emotion that was felt. And we use this protocol on around 38 healthy subjects in the psychiatry unit of Polytechnic. We have published this protocol because it was so difficult to be implemented. So when I wrote the project, I thought it was like I was reading potentially. And then when we needed then to set up the protocol, we took a long time to refine it. So it's published now. And then finally, we had fun with the data. So we analyzed the EEG during the VR navigation by using power and entropy measures. We used the connectivity through frequency-based connectivity. We also use the EEG microstates that are like sort of transients of brain activity patterns over time tracked through the EEG. And this is the work of the PhD of Flavia Carbone here in Politecnico. And we have the TMS EEG potentials, that track the excitability of the cortex after the VR navigation. And this was also like studied with respect to the subjective emotional experience. So here is just an example of the, let's say, power in the different frequency bands during the navigation of a space scenario that was inducing the O with respect to the neutral one. So we have an increase of the occipital power in the delta, theta, alpha, and gamma bands. And we found an increase also of the entropy in the central regions in gamma and alpha. And we found also that the main patterns of macro activation captured through the EEG microstates changed a bit during the O experiences compared to the neutral ones. And mainly we found that there was a disappearance of the default mode network activation during the O experiences. That is something that was hypothesized in our work. And using the TMS EEG evoked potential, we also found that the amplitude of the fiducial peaks within the TMS EEG potential was proportional and was greater after the VR scenarios that were inducing the sublime compared to the reference one. So we hypothesized that there could be an increase of excitability within the dorsolateral prefrontal cortex after the navigation in these scenarios that were inducing this specific emotion. And this was also found here. We found that the intensity of the experienced emotion was like proportional to the amplitude of the peak N100. So, and the same is here. We found that like these are the 3 O-inducing scenarios. We had greater latency in this case of the P90 parameter. So these are just preliminary data, but we are still analyzing them. I don't know how much time I have left. I already finished. Okay.
No problem.
So maybe I do just like a quick overview of the multi-site data integration that we are performing. So we move to the other, let's say, side, and we go to the sample size and numerosity, let's say, side. So we have we have some research activities in collaboration with research networks in Europe and outside Europe. And I will show you an example, just one, of the stratific network that is a network that we have created with Professor Brambilla, where we have collected data from different centers, that is magnetic resonance imaging data from healthy controls and bipolar disorder patients. So these patients have this bipolar disorder, and we wanted somehow to use this data to provide some personalized medicine information for the management of bipolar disorder. And with Ines Sampaio, who is a PhD student here in Politecnico, we have used like a sort of deep learning framework of normative modeling and anomaly detection to try to somehow learn the normality and the like reference brain structure. And we use this reference and normative model then for the data set of controls and bipolar disorder patients to extract the individual level deviations within the brain in bipolar disorder individuals compared to the healthy controls. And to do so, we use the training sample, the Human Connectome Project dataset MRI dataset that was of young adults, around 1,000 young adults. And then we use a same age sub-sample from the Strativip network of around 350 healthy controls and almost 200 bipolar disorder patients as a test sample. So we extracted cortical thickness and gray matter volumes and white matter volumes from regions of interest, and we put them in the normative model estimation. So we estimated the norm of these volume-based and surface-based features. And then we applied this model. We used it on the Strativip test set to extract the reconstruction errors of bipolar disorder with respect to health controls within the Strativip data set. We used an autoencoder that you will know more than a lot what it is. So we used it to try to learn a latent space that enables the perfect reconstruction of the brain morphological features. And this autoencoder was trained on the Human Connectome Project training dataset. And then after having trained this model, we gave as input data from unknown subjects from the test set, extracted the reconstruction error, and tried to understand more of the individual level deviations and the group level deviations in bipolar disorder. This is the framework that was quite complex because it also included a part of multi-site data organization because we have multiple sites and data is very inhomogeneous across sites. And then we performed, after having like done all the confounding correction strategy, we performed the normative models, and then we performed different types of statistical analysis. And for instance, statistical analysis were performed both at the individual level and at the group level. And I will show you some examples of individual cortical thickness deviation maps. So we could extract statistically significant deviations at the individual level compared to the normative population. And in this case, we have the individual deviations in one healthy control versus one BD subject. And using this framework, we also extracted the extreme deviations. So we extracted those subjects from the test set that was outside of the 99th percentile threshold of the normative distribution. And so we could mark somehow the extreme deviations of single individuals and single features. And we found that the extreme deviations in bipolar disorder were many in some specific features, like the white matter volumes and gray matter volumes of subcortical regions, were more prevalent than in healthy controls. And this was published here. We have very complex the methodology, so if you are interested, we also are available. Ines is available for any collaboration or clarification. And so I will probably stop here because it's too much. This is quite long. So we have another work on the same cohort, the extractive dataset, where we have used GNN. And in this case, but I will not show you probably it's just to show you. If you are interested, then we can of course speak of it as well. So just to recap, we have an overview of the cognitive technology and analysis tools for the study of the human brain, both in physiology and in pathology. We found that there are multiple modalities that when are integrated, they can really give like an added value. And also the pipelines for integration and multimodal and multicentric brain features are very relevant in this context. And of course, we suggest at the end that we want to have multiple spatial and temporal scales, multiple acquisition techniques, but also multiple analysis techniques of the same datasets. And of course, we need the more standardized approaches to enhance reproducibility. And I thank you so much for the attention. And here are some, let's say, links to dissemination material. I would like to thank the Brain Lab at the Polyclinic of Milan and the Bicupo Lab and ELF Lab here at Polytechnic of Milan. And thank you so much.
Thank you so much for
I will leave you probably. Thank you.
If there are questions from the audience or from Marcello is not here anymore, but if you have any questions, we are available.
Maybe I will take out of an hour with questions, but I will go maybe short with just one. I think that my research is somehow complementary because you focus a lot on the like brain imaging techniques to see what happens actually in the brain. My research instead tries to focus on some maybe signals that can be collected in sort of more ecological way to provide some tools for support diagnosis that of course don't want to be diagnostic tool. You mentioned bipolars and depressive.
Yeah.
I have been working on a dataset that starts from cognitive tasks. It's basically GoNoGo, so the one you mentioned. And we are actually struggling a lot because it's not that easy to find features that can be somehow like an indicator of depressed versus bipolars versus affective controls. So I was wondering whether, according to the studies and the brain activation that you mentioned, there could be an hint or an idea of what we could focus on, taking into account that we only have the cognitive task actually. So it's not that easy to understand the features to like focus on and to understand how to maybe extract some sort of, let's say, biomarker, but it's not different what I know for this task because we're struggling on that.
And you have the EEG as well?
No,
or
not in this task.
Cognitive performance.
Yes.
So you, of course, you will have already extracted like the response times and they don't change.
Yes, basically you can have the 60-70% accuracy in like discriminating the bipolars versus the depressed versus controls. But I was wondering whether there could be maybe something in the brain activation that can be reflected in the actual performance you do that can be somehow help in performing this task because like somehow when you want to go just in an ecological context, it's not that easy to have all the other signals, but then the task becomes difficult. So
yes, of course. I don't know because in an example, one of the tasks, it's true that the parameters that are taken are response times, accuracy of identification and inhibition, like effectiveness in the inhibition versus the target, correct target identification. Maybe the only way to extract maybe some further information could be to like create a different experimental protocol where you have also some attention shift. This is an attention shift task, but it could be that what I know from my research, let's say I'm not clinician, but I know that they have very difficulty in engaging in attention shift tasks because they have a rumination that is like depressed individuals. They have some challenges in engaging in cognitive tasks because they have a lot of self-ruminative processes. So maybe what could be if you want to enhance the identification and emphasize the identification, you could perform something before that could focus more the participants on their own problems, and then maybe they have more difficulty in getting engaged in the following cognitive. Maybe I would do something like that in terms of cognitive protocols.
So, like starting from your experience in integrating data, so the idea could be before starting with some integration that can make us correlate, like maybe in the actual performance with some other biomarkers that is making more informative
value. Yes.
So
these are like behavioral tests. So maybe I would do something like questionnaires that maybe can let's say elicit their self-domination.
Okay.
And if you want to emphasize the differences in the specific task, that at the end will just have or use emotions in the task. So we have a sort of emotional continuous performance test that I would not suggest go for because it's a mess like the analysis of. I would not suggest it. But if you want to track the performances, those maybe could be, because if you have an emotional balance that is related to the target and non-target stimuli, at that point we have an emotional continuous performance test that is exactly equitable, where the subject had also to focus and to discriminate emotion, a target emotion. Like we had a positive block where the subject had to press only when faces were with a positive valence or negative blocks and so on. And maybe that could engage more their affective, let's say, but it's not easy. But you are also very expert of wearables, no? So maybe you can put even just 4 electrons, as Leonardo was saying, could be enough.
Yes, like in some projects we focus on also like the fact of having like the complete EEG and then try to reduce. So what if we only had, let's try to simulate we only had 4 channels, that is some sort of wearable setup.
Yes.
While on the other task, like I'm exploring also like collaboration with some sort. It's basically a keyboard that you put on your phone and tries to track mood disorders starting just from the way you type. That could be great because you can really insert this assessment in an ecological setup, but of course it completely lacks the physiological information I'm used to work on, and so it's not easy to understand.
Yes. With the smartwatches now, people are tracking their own. So maybe that would be a good.
Yes, and useful integration.
Yes.
If there are no further questions, I will thank you again for the talk. It's a pleasure.
Sorry for the many things. I hope.
Thanks for the audience for participating. We will see you next week with a new Friday talk from Laura. Thanks again. Have a good day.
Thank you so much. And I'm available for any further questions by email.
Thank you.
Bye.